Hi, my name is Hirak. I work with Professor Raphael as a Ludwig Scholar at Princeton. I'm extremely grateful to be here and present my work. Uh, so, uh, this will be on cell-cell interaction in spatial data, and we try to develop a new statistical model for doing an accurate interaction prediction and describe the model. Uh, so, cell-cell interaction between the nearby cells had been a central feature of cellular biology. That's how cells talk to each other, and a range of different biological functions are driven by such interaction, and it's been well studied. Although the quantification of such interaction had been uh, a mystery because we did not have the correct resolution except looking at a set of genes or antibodies via staining or so on. And very recently, we have gained a lot of information from spatial data. So one of the ways that we used to estimate cell-cell interaction is through statistical estimate of correlation coefficient, which is often a point estimate. And what we get from here is basically how they co-vary with each other. Now, one problem for such a techniques, which had been age-old in computational biology, is that it relies on individual expression of a ligand and a receptor in single cell. The problem for such interaction predictions in single cell is that this is all an approximation in some sense because we don't know which cells are really nearby. So, except when the cells are not very close by, it's very hard to understand that the interaction is really happening or if it's a statistical uh, um, estimate which is not based on actual interactions, but still the actual interaction is hard to predict. As we would see that recently with spatial transcriptomics, we have this idea of which cells could be nearby. Although this has given us tremendous opportunity to look at the cells which are close by, but still we have no idea. We're still challenged with low data, which means that low throughput, gene coverage, read depth, and so on. So spatial data is still in its nascent state, where that means that it's not completely evolved to give us as high throughput as bulk RNA-seq counterparts. Now, how much you would like to look at the cell-cell interaction as correlation coefficient? When we look at the actual count data, it doesn't look that well, which means that individual spots often only expresses a handful of genes, although it has a huge gene panel, but that doesn't always mean that the individual ligands and receptors would be very expressive, which would lead us to modeling such data, uh, which would be more difficult. And so when we look at, say, for example, a very well used spatial data set, for example, 10x genomics VZM data set, you'd see that the medium UMI count is kind of high. But when you look at, for example, this is a Frizzle receptor example, which is a median of expressed receptor and medium ligand. This scatter plot shows that we still have kind of sparse expression, and that lead to many problems, as we will see. So when I first started this project, one idea that I first tried as a computational person is to model this as a bivariate count data. And bivariate count data is not that well studied in CS, but in stat, it had been quite a well studied thing for many years. And what I just realized is that there is no canonical way of modeling bivariate count data. So, for example, if you look at bivariate Poisson distributions, bivariate negative binomial distributions, those distributions have many bottlenecks. Inference is very difficult. For example, in bivariate Poisson distribution, the range of correlation coefficient that it models varies from 0 to 1. So the negative correlation coefficient is not modeled in some of the cases. And so we looked into more sophisticated versions, and I found something which is known as copula, which is very well known in statistics, but interestingly not so well known in CS community. And I started looking at this literature of understanding how you can take arbitrary distributions and couple them. And so the couple is the word from where this notion of copula arises. I know that within this talk, it would be hard for me to describe copula in its entirety, but I would try to paint the way that this thing works. So what happens is that you start with any marginal distribution. So for example, you can start with Poisson distributions and say you have two variables, and you want to understand uh, how their correlation changes, or how the covariance, or model the covariance structure. And what happens is that you can take the cumulative distribution function for individual marginal distribution. So for example, if you have Poisson, you can take the CDF of Poisson distribution and convert them by distributional transformation, and then can apply inverse CDF of the coupling distribution that you want to capture. So for example, in this case, see, you want to model this bivariate count distribution in a way so that their covariance is modeled as a normal distribution. So you use a Gaussian copula and you use inverse CDFs from Poisson. So this gives you a lot of liberty in modeling distributions which are not as well behaved as Gaussian. So for example, you can have on one hand negative binomial in one variable, you can have Poisson on the other, and you can still model their covariance. So basically, this is what the copula technique is all about. Uh, so we try to look into that. What are the benefits that copula gives us? So first of all, it removes the distributional assumptions that something like Pearson might have. And so on the other hand, it also do not have the problems that bivariate count distributions have, which means that it can model a full range of covariance. And on the other hand, it's there are ways how we can make inference on such probability structure. So basically, you can adapt this model. This is the simplest version of copula that we can apply in ligand-receptor interactions. For example, in case of spatial data set, what one can do is that they can make a spatial proximity graph, and within the spatial proximity graph, the ligands and receptors, they interact pairwise when they are in spatial proximity. And then we can use marginal distributions and Gaussian copula, and then run a maximum likelihood estimate to infer the covariance structure, basically. And this gives us many interesting features. One is that since this is a spatial data set, uh, we can estimate a background distribution by permuting the spatial points, and can also analyze significance of the uh, of the detected interaction. There is one problem with spatial data sets. We don't have really good ground truth. So, of course, being a computational person, I first tried it on simulation, and we have tried many different distributions. And the good thing about copula is that since you can really play with the marginal, you can make the data as sparse as you want, and you can see that how copula correlation coefficients are less sensitive to sparsity. So for example, here the x-axis tells us the percentage of zeros. And what we try to show here is that when you have enough data, the correlation coefficients agree with each other. But when the data is sparse, sometimes it's problematic for uh, existing correlation coefficient methods that are very very well used and well known. They are not that applicable for sparse data sets. uh, on real data, what we have seen is that when we compare with other methods which use similar techniques for correlation coefficient in different shape and form, it gives different results. It's hard to understand that what if the interaction is really happening or not. So what we try to do is that we try to give a significant score via spatial permutations. So it's basically a bunch of spatial spots could be randomized, and then one can build a background distribution and see where the estimated correlation coefficient from copula or such method would be significantly high or not. The other thing is that since it's a probabilistic model, one can think about assigning a score to interactions between the nearby spot. In this case, uh, uh, I do it with Mahalanobis distance, which actually just measures that where the if the score is away from the mean. And we can highlight some regions in spatial spots. So in this data set, for example, we found the chemokine receptors, which are otherwise lowly expressed and therefore hard to detect. These interactions are between tumor and surrounding tumor for this data set. They're very well supported in literature. Unfortunately, it's not a ground truth, but it's biological sort of coherence. Uh, we also tried on a different data set from VZM, which is again sparse. So for example, here it's on Sickfish Plus data set where the gene panel is smaller than VZM, but the sparsity exists. And our tool found a series of want receptors, which are also kind of well known in such space. Uh, to summarize, this is implemented in Python, and the tool is known as Copulachi. What it does is that it can take in a spatial data set. It can find from one cell type to another cell type. It can produce the interaction score and assign a p-value. And hopefully on sparser data sets, it would perform better than existing methods, which relies on distributional assumptions uh, when it comes to count data set. We right now are working on some variations of copula where, since this is a probabilistic structure, we can incorporate many parameters. For example, one can introduce the differential interaction parameter to measure or take two regions within a spatial data set and can find out if one region is differentially more expressed than the other. The software is available from this QR code, and I want to thank my advisor Ben and my co-author Utsa, and also Ludwig Institute for supporting my travel to here. And thank you all.
Maybe I can just get started myself. The data type that you use, let's say Visium versus different type of spatial data, and the type of copula that you use, are they somehow connected? And whether that's a factor that you have to consider when designing
Right,
the model?
that's a great question. So for if the data is very, very sparse, then one can think about using GLM-PC or those kind of things, which are a slight variation of Poisson distribution that can account for total UMI on a spot. Because otherwise, if you do not control for the total UMI, the sparsity might not be handled. One can also think about using inflated negative binomial distributions. What I saw in Coppola is that the best thing about it is this kind of flexibility. But yeah, so definitely there is a relation. Yeah.
Thank you.
So I just wanted to ask a clarifying questions about the CXCL13 and with the ligand. So what you're detecting is a correlation between the ligand and receptor in gene expression, but the cell-cell interaction may actually be causing other transcriptomic changes, not actually due to correlation of expression. So
Right.
I'm just curious.
That's not actually at all included in the model. That's a great question. I feel that here my concentration was mostly on the side of correlation coefficient and how that happens. But to be very honest, if one wants to be extremely uh, uh, particular about detecting biological interaction, I feel that probably biophysical models are better, or models which also simultaneously look at the downstream gene expression might be more effective. So, for example, in my opinion, COMOT try to model that, which is a OT based tool, optimal transport based tool, worked really well. Yeah, yeah, but this model doesn't income.
Cool, thank you.